This brings us to, to my, I don't know, personal favorite, but the one that I worked on in my second postdoc, which is complex 3. Complex 3 also has a couple of different names, right? Another name that they give for it sometimes is called the bc1 complex, and that's in lowercase, technically italics. So just in case I refer to it as this, it's also called the bc1 complex. And that's because of the hemes and cytochromes that it has inside it. So complex 3 is huge, and it is a dimer of polymers. There are multiple proteins that all come together to form one portion and one big unit, and those two big units come together to form a supracomplex. And so that's why we have this one portion that they call the risky protein. This is the risky protein, and it has a 2Fe, 2S iron sulfur center, similar to what we've seen in the past, that accepts electrons. But the risky protein of one big complex, actually takes the electrons off of the other big complex that it's dimerized to. So that's why I mean it's huge. We're going to talk about the way the electrons flow in just a moment. But if you notice, remember we also talked about the p side, which is the positive side, where more protons are, versus the negative or matrix side, where it has the lack or the dearth of protons due to the proton pump. It's not obvious here, not shown here. Whoa, but there are there are protons that do traverse the membrane, move from one side to the other during this process. Okay, some of the other things to point out is that there are two hemes. The L and the H stands for their electronic potential. So this is heme B with a low electronic potential. This is heme B with the high electronic potential. Then we have a heme C1, which is near the P side on a different protein of the complex. And then this is where it gets kind of tricky. Cytochrome C is a separate protein altogether with its own cytochrome. Don't confuse it with cytochrome C1, which is part of the BC1 complex. So I wish, personally, I would have liked to have given them all different names and stuff, but they didn't. They were very redundant. The Q cycle occurs right through here, and we'll talk about what it is in just a moment. I've got to bring it up on the computer so that way I don't overlook anything. Okay. I absolutely dislike this figure because it took me a long time to understand what the book was trying to say here. Okay, this is the Q cycle. And I told you, it's a doozy. Where'd I put my soda? I'm going to try to walk you through it. And starting to battle a sore throat. Remember, the coenzyme Q has three states. There's the ubiquinol, which is fully reduced. There's ubiquinone, which is fully oxidized, and then that semiquinone state, which had one electron, so technically it's a radical. That semiquinone state is the one that's got that Q dot minus. Q, when it's just the Q, means that it's oxidized. When it's got QH2, it's reduced. So let me explain. I'm serious. It took me forever to figure this out. It seemed like forever.
You said the dot Q9 is semiquinone?
Yeah, this is semiquinone. That means it has accepted one electron and technically one proton. But it is so this would be the oxidized, which lost the electrons. This is fully reduced, where it's gained both electrons from either complex 2 or complex 1. Now, let me explain one other thing to you. The way that this is set up, these are two different complex 3s. Because remember, I mentioned this briefly in the sense that all of this is occurring so fast that a lot of these complexes kind of gather together, and I I think they're called like respirazones, or where these big complexes of complexes to where they are physically located close to each other. There are 2 binding sites for every one of these for Q. And so a lot of times they'll call it the one that's on the P side and one that's on the N side. So that's what this is trying to show you. Here we have one where one Q is bound, and here's the little, I'm going to call it lower, but the N side Q. See, when I say N side, it means I'm saying inside. Negative side Q. I hate saying lower and higher, because I'm not going to, because it's the opposite electron potential. Okay, so I'm going to have to say P side and the letter N side. At any point in time, if you're healthy, you have both sites will be bound with a Q of some sort. That's important to know. That's why you're constantly having this Q cycle going. All right, so let's imagine this. We have a QH2 that's coming from either complex 2 or complex 1, it doesn't matter. So it's got 2 electrons that it wants to get rid of, as well as 2 hydrogens. It moves in to the P side position. The P side position always has a QH2 coming into it. Now what's interesting, and remember one characteristic of Q is that it can give off electrons one at a time. So at the other site, the one that's on the letter N side, maybe if I just put space there, N side, there is always going to be either a Q or a semiquinone, the Q dot. It's one or the other. And I'll explain the reason why in just a moment. Okay. So that's important to know. There's always 2 types of Q bound. And remember, Q can donate 1 electron at a time. So here we go. We've got this QH2 coming in from complex 1 or complex 2. It comes in to the P side. What happens is 1 electron, what they've done is this is part of the risky protein, the one that looks like a hammerhead. They deleted the part that's not as important, the part that moves from spot to spot. Okay. So what happens, let me change colors. 1 electron from the QH2 goes up to the risky protein, to that 2Fe2S center, because it can accept. Then that protein moves, the head, so it looks like the piano, the risky head, takes that 1 electron and gives it up to heme C1 of the cytochrome C1. Then that takes that 1 electron and gives it to cytochrome C. Cytochrome C can only accept 1 electron at a time. It's not like Q. Q that can do 2. This cytochrome C, now it's reduced. It can go on to complex 4, and it's happy. But we can't leave this alone because there's still, let me change colors. I don't want to use that color though. Let's see if this color shows up. We still have that 2nd electron. Remember, we only got rid of 1. We've got a 2nd electron. What the Q cycle does is that 2nd electron goes, I'm going to call it down, because it's down an electron potential gradient. It gets passed off towards the mitochondrial matrix side, that N side, to first the heme BL, because it's got the lower potential. Then it goes to heme BH, because it's got a higher potential. Then it goes down here to that 2nd Q site. Now, if it was a Q that was fully oxidized, it gains 1 electron and it becomes semiquinone. So that's what that's trying to show here, is it gained 1 electron and it goes over right there. The other option is this right here. Notice the top is identical to what we did before. But on the bottom one, that 2nd electron went to BL to BH, but instead of it going to a Q, it goes to a semiquinone. Okay? And so that's why it depends on if it's a semiquinone right here or if it's a Q that flew in from before. Now I'm going to go back up to the top. I know it kind of goes all over. Now, since we've got rid of both of these electrons, it's no longer QH2, it's just a regular Q. And that Q can go back to complex 1 or complex 2 to gain 2 more electrons. Or that Q can go down here if this spot is open and be available to accept electrons. That's why it's a cycle. And so you never short circuit out. You constantly have a pool of Q and a pool of QH2. We do have that semiquinone that, if you notice, it's either going to be already waiting down there or it gets just moved over to the neighboring spot and be ready to accept it from its neighbor. Okay, so then what happens is if we start off with a semiquinone down there, whenever it gains that second electron, it picks up 2 of the protons and becomes QH2, and it can leave and travel up to the top spot. So that way you don't ever short circuit your battery. You constantly are replenishing the electrons, and then the protons by default are going outside. Because if you notice the first 2, I call it the first 2 went out, and then we picked up 2 more from the inside down here to make this QH2, which ultimately will go out as well. So that's why it's called the Q cycle. It takes a little while. This is the electron, like if you remember trying to do those electron balancing redox reactions from Gen Chem. That's what this is trying to show you. If your mind doesn't, and that's why it shows you that overall when you add them up together, you have QH2, you have 2 cytochrome C1s, because for every QH2, it's going to happen. For the entire Q cycle, you're going to go through do it twice. 2 more protons that go from the N side that goes to make 4 going to the outside. Okay, then we get a regular Q, and then you get the reduced. And we'll talk about what cytochrome C does. Is remember, it jumps. It's the only part that stays in the intermembrane space. Unless you're dying, unless you're dying, unless the cell's dying or something's wrong, right? And then it can signal for apoptosis. Okay, you know, it's a lot. It's a lot. Let me, because I don't think it'll let me erase everything to go over one more time. So let me see if it'll let me do it this way. Okay, I think this is a beautiful picture. Kind of reminds me of that what Bush's big bean commercial. Roll that beautiful bean footage. All right, so you can kind of see the overall scheme, like how everything happens. It's lovely. It's beautiful. Okay, so this is where the crystal structures. Some of them, sorry about that. Some of it is unknown. You know, the part that I call the panhandle is unknown where the putative Q cycle occurs. If you start off with NADH, becomes NAD. We can see how it runs up through complex 1. This is where Q becomes QH2. They just show it's Q. It shows it doesn't actually leave the membrane. It actually goes around complex 2. It just doesn't go through complex 2 to complex 3. Likewise, we have succinate becoming fumarate. That's from the citric acid cycle. But it does so by FAD becoming FADH2 inside here. And so then FADH2 then passes it all off to coenzyme Q as well. Coenzyme Q then comes over here to the complex 3, which you can see is a huge complex. This is where the Q cycle occurs. 1/2 of the electrons will flow down to either oxidized Q or semiquinone. The other one will flow up through the risky protein, which you can't really see here, but will flow up to cytochrome C1, which will give it off to cytochrome C, which flies over here and gets oxidized by complex 4, which ultimately make water. We can see that protons get pumped across complexes 1, 3, and 4. So that's kind of how everything flows in and fits together. Are there any questions over this? This is the kind of thing that I was talking about, how we were going to put it all in. The only thing that's missing here is we don't show the ATP synthase, which is the oxidative phosphorylation portion. All right. All right, so now we have oxidative phosphorylation. Oh, now I can't get it to go even. This is where all of those protons that we've been pumping across into the intermembrane space, this is why they're important, and how they're going to ultimately go through the little turnstile and cause the ATP to be made. And we already showed the video last class about how that happened on Friday. This little thing that kind of looks like a ratchet. It's called the stator protein. Stator protein. And then we have the sometimes called the F0, the axle, and the F1. I think that's based off of the E. coli nomenclature. It's not that important. But once again, the protons are going to flow back through into the matrix through the channels down here. It's like a little turnstile. And by doing so, it's going to cause this to go rotate around and around. And at any one point in time, you're going to have... Do I not have it at all? Oh, I don't have one that shows it. So I'll draw it. You're going to have what's called, some books call them A, P, and O. Some of them call them A, P, and E. That letter doesn't really matter. It's just a way to remember what's going on. So this is what it looks like. If I take I did the wrong one again, sorry. If I take this and I turn it on its mushroom head, to where we're looking down what looks like the mushroom head, where they've got the two different colors of, is that pink or red? One of those is called the alpha, one's called the beta. This is what I was kind of hoping that I had put down. So at any point in time, maybe it would have helped if I would have drawn the circle first, the mushroom head. There's one, and I can't remember if your book calls it A, P, and E, or A, P, and O. The E may be an O for empty or open. Okay. It's just one way it's easy to remember. And so what it is at one point in time, the binding site at this alpha beta interface for the A would have an already previously made ATP in it. The P site would have ADP plus an inorganic phosphate. And then the E site is empty or open. Okay, that's why it's either E or O. It doesn't really matter. It's empty or open. There's nothing there. Then what happens is as the protons move through, it causes like a 60 degree conformational change. This time I'm going to draw the circle first. And previously, what had been the ATP, since it's already fully made, it leaves. And so now that site is the empty or the open site. Okay. And what had been the ADP plus inorganic phosphate is now ATP because it was literally smashed together. And what had been empty or open, since it was open, that's where the ADP and the inorganic phosphate come in together. And so that's why we essentially, what had been a P is A. Whoops, that's supposed to be A. This is P. And once again, I think you can see where this is going. We have more protons that flow through the turnstile. The one that had been empty is where the ADP and the inorganic phosphate have go into it, and so it's the piece. Oh, whoops, I didn't change my color, sorry. So it's the P site. The one that had been ATP, the ATP left, and so it's now the empty or the open site. And the one that had the ADP and the inorganic phosphate from previously was forced together to make ATP, and so it's now the A site. So that's why it literally turns it and cranks it and forces the ADP and the PI together. This is just a little tiny cartoon showing the same thing. In case you're having problems visualizing it, or in case the crystal structures like overwhelm you and you want something that kind of looks like it should belong like Super Mario Brothers. Not that I'm endorsing Super Mario Brothers. Please, no product endorsements yet. Maybe that's one way I can make money. Like have a little Mario or something like jumping around. Okay, respirazomes, like I told you before, in chemistry of cancer, we've talked about this a lot. You know, biologists and biochemists, we like those zones and those ohms. And this is just where, lo and behold, you can imagine there's so many of these complexes. The inner membrane of the mitochondria is protein heavy. There's lots of proteins because there's an electron transport chain occurring. And it just makes sense that they're going to kind of come together and be in close proximity, so that way coenzyme Q and cytochrome C don't have to travel very far. And you can kind of see these spaceship-looking things together using different forms of microscopy. And so that's why it just shows you that it's not far apart from complex 1 is not that far apart from complex 3, and so on and so forth, because of the fact that they kind of co-localize within the same region of the membrane. And there are lots of them. Okay, so now we have to talk about our shuttles. I don't know, it doesn't come across very well, does it? There are little school buses in the background there. You know, the shuttle bus. I try to be cute once in a while. My wife says I am, at least. Bada bing, I'll be here all week. See if you guys were awake. So, we've been talking about a lot this semester, and we've been making NADH or FADH2, and it's pretty obvious how the FADH2 from the citric acid cycle enters the electron transport chain, because it really doesn't leave. Succinate dehydrogenase is a part of the electron transport chain. One problem, though, is, once again, even if you don't look at the screen, and I've been trying to stress this for compartmentalization, where does glycolysis occur? Cytosol. So we somehow got to get that NADH, which we made NADHs, from the cytosol, and we also make NADHs from other things too, right? But we get those NADH from the cytosol to the inner membrane, and not only that, but to the matrix, and come to find out, NADH does not pass through. That's a holy of holies. The mitochondrial matrix is so difficult to get into for many things. So we have to use these shuttle mechanisms that, in effect, move electrons, because we can't just throw the electrons at the membrane, because otherwise bad things would happen. And so they use shuttle mechanisms, and there are two that are important. Well, I guess they're all important, but there are two important ones. The first one we're going to talk about is called glycerol phosphate, which this is something that's, I think, is brand new. We haven't really talked about this. Remember, glycerol is one of the few compounds I said that by the end of the semester you're going to actually know its structure. So if I asked you to draw glycerol phosphate, by this point in time, I would expect you to know how. What is glycerol? How many carbons does it have? 3. And what's hanging off of every single carbon? Hydroxyl group, an alcohol. And what a glycerol phosphate is one of those. One of the terminal ones, like one, you can call it carbon 1 or carbon 3, because it's symmetrical. It is as a phosphate instead of an alcohol. All right, so glycolysis, cytosol, NADH. Nothing new, nothing earth-shattering. Hopefully it's not new for you. Otherwise, go back and look at what we've been doing this semester. NADH cannot cross the membranes. However, glycerol phosphate and dihydroxyacetone phosphate can. What was a nickname that we like to call dihydroxyacetone phosphate? DHAP. That's our good friend DHAP. And where have we seen DHAP before? It's also glycolysis. So we have glycerol phosphate and DHAP that can go back and forth. So, in doing so, we can take an NADH from the cytosol, and we can take its electron, so it's going to become an NAD plus in the cytosol. But then when we transfer it over, if you notice, only 1.5 ATP gets made because it's an FAD on the mitochondrial side that takes those electrons back. And so whenever it becomes an FADH2, that means it starts with complex 2. Now, if there's some way to do NADH before, then it would be 2.5. This is just a little side note. Originally they found it in insect muscle, but lo and behold, it's also in our brain and our muscle as well. So this is what it looks like. I'll give you a moment to get caught. So this is why I told you, if you knew the structure of glycerol, 1, 2, 3 alcohols. You just put a phosphate instead of that third alcohol, and you have glycerol phosphate. At this point in time, you should also be able to draw a DHAP, because you know from organic chemistry what acetone looks like. Acetone is a methyl group, carbonyl, and a methyl group. Dihydroxyacetone, lo and behold, is just where those two methyls have alcohols hanging off of them, and then you add the phosphate. So don't get overwhelmed by just the name. Just try to think it through logically. And, well, it's not real. I would have written it here. Cytosol. Cytosol. It's written up at the top, but I'm just trying to make it really obvious here. Cytosol. Okay, so we have DHAP. The DHAP in this instance comes from one of two places. We get it from glycolysis, but it can also come from the inside of the mitochondria because it can go back and forth through the transporter there. What's the only difference between DHAP and glycerol phosphate? Right, we have a carbonyl here, and what is it in glycerol phosphate? It's a hydroxyl, an OH. So what kind of and notice we have an NADH. This is the NADH that we've been making from glycolysis, or we made it from something else, but it cannot go directly in here, right? We want those electrons. We've got to get those electrons to make energy. This is how we can effectively do it. So what kind of enzyme or enzymes can take something like this, a ketone essentially, make it to an alcohol at the expense of an NADH becoming an NAD? Is a dehydrogenase. Okay? So if you were going to name it, there are two possible names for this dehydrogenase. What's one of the possible names? And this one is glycerol. The other one would have been DHAP, by the way, but this is glycerol phosphate dehydrogenase. See? I'm gonna. I don't have on the next slide. I've got it. It's all typed out, but I just don't have enough room. Now the glycerol phosphate can just go ahead and move on into the mitochondria. But here, if you notice, it's an FAD that becomes an FADH2. It's not NAD. But we have glycerol phosphate becoming DHAP. What class or classes of enzymes can do this kind of reaction, alcohol to ketone, using an FAD? As a dehydrogenase. Yep. This is also dehydrogenase. If you were going to name it, what would you name it? You could, but this is also glyceraldehyde. Glycerol phosphate dehydrogenase. The only difference is this one's called cytosolic, and it uses NADH. This one's called mitochondrial. Let me just go to the next one that's written out for you there. The mitochondrial isoform utilizes FADH2. That FADH2 goes where in the electron transport chain? Complex 2. Okay. And I've really been trying to take my time to go over this chapter because